think it's time for us. Since Dr. Shan Sharif is busy in a meeting today, so I'm having the pleasure to kickstart the seminar. And I would like to welcome you all for our second PHRN seminar in the 2016 and 17 academic year. And I would like to also welcome those who are joining us through the webinar. So before I introduce our speaker, I would like to share with you really a great news which was shared by Dr. Sharif in the last previous seminar. And I thought, for those who don't know, I should tell you that we are really delighted to have received a generous gift from the Parks family. So the PHRN has received this gift. The Parks family has a strong family history with OVC, including Dr. Vic Parks, OVC 64, Dr. Geraldine Parks Clements, OVC 88, Dr. Victoria O'Leary, OVC 94. So it's also interesting to note that Dr. Victoria O'Leary is the Chief Poultry Veterinarian for the Government of Newfoundland. So on behalf of PHRN, I really thank this generous gift and also request you for a round of applause for this family. Okay, so our today's speaker is Dr. Suresh Neethirajan. I bet I'm the only one in the room who can pronounce his last name correctly because we come from the same place, from the southern India. I have a long list of accomplishments and achievements about Suresh, but I have tried to make it as brief as possible in a bulleted list. So Dr. Suresh Neethirajan is currently a registered professional engineer with the province of Ontario, and he's an assistant professor in the biological and biomedical engineering program at the University of Guelph. Many of you might have heard about the Bio-Nano lab down in the School of Engineering, and he's heading the lab as the director of the Bio-Nano laboratory. He did his PhD and master's from the University of Manitoba, and he joined Guelph in 2011. Before joining Guelph, he worked as a research engineer at the Oak Ridge National Laboratory of United States, as well as a research scientist in Japan at the National Food Research Institute. In 2010, there is quite a bit of list of awards that he has received during his career. In 2010, he was recognized by NSERC Canada with the National Nano Innovation Platform Award, and in 2014, he received the Early Researcher Award by the Ontario Ministry for Research and Innovation. And in 2015, as many of you might have noted from the UFG website, he received the Young Engineer Achievement Award by the Engineers Canada for his research excellence and contributions. So he is also a member of Canada's National Nanotechnology Standards Committee and the Vice Chair of the Subcommittee of Emerging Information Systems. Interestingly, Suresh has been invited by the Parliament of Canada as an expert witness for enacting the Bill S11. I have no clue about that bill. Suresh might have to tell us regarding the standards for nanotechnology in agriculture. Suresh, so far in the last 4 years, has been very successful in gathering $1.8 million of funding, and he has published several papers, 150 technical papers, including 60 peer-reviewed, international high-impact journal papers, 5 book chapters, and he has 4 US patents. And he has many MSc and postdocs under his supervision, and he also teaches bioinstrumentation design and biological systems at the School of Engineering, University of Guelph. I'm there. So take it from here. Thanks, Suresh.
Thank you, Ravi. Thank you, everyone, for being here, being a post-Happy Thanksgiving Day. Thank you also to Shah and Sharif and also to the Poultry Health Research Network for giving this opportunity to connect with you to talk about some of the examples of the work we have done in our lab and also have a glimpse about the potential of bioinstrumentation and biosensors that can bring novel solutions for practical issues in the poultry health industry. Just a bit about a 1-minute video clip about what we do and who we are. I think probably I will run this from the
Located at the University of Guelph in the Canadian province of Ontario, about an hour from Toronto, is the Bio-Nanolab. Researchers at the Bio-Nanolab help keep our food supply safe by improving the way farm animals are diagnosed and treated for health-threatening symptoms such as ketosis and avian flu. They develop new tools by combining biology, chemistry, and physics with tiny mechanical structures known as bio-nanotechnology. Unhealthy farms not only threaten the lives of livestock, but also the lives of humans. So, Dr. Suresh Neethirajan and his team of researchers study the complex behavior of molecular cell mechanisms in order to develop new tools that benefit the agricultural sector. Their research of nanoscale biological systems has led to the development of analytical and diagnostic handheld tools that can emulate a lab in the field. This means animals with health issues can be diagnosed on the spot without waiting hours or days for a test result. The sooner a diagnosis, the quicker a remedy. No lab time also means savings for the producer. So far, the Bio-Nanolab has developed a biosensor for detecting the cause of milk production in dairy cattle, early identification of avian influenza in poultry from cell structure behavior, chromosome imaging that marks genetic disorders, methods of screening pharmaceuticals for their effects on pathogenic bacteria, a microfluidic tissue model that emulates wounds and allows treatment in cases of drug resistance, and a biosensor that quickly detects allergens like peanuts and gluten. And many of these developments are currently being used in the field today. The Bio-Nanolab has also been researching ways to prevent harmful bacteria from infecting wounds and prevent rejection of implants by using an electric current. The Bio-Nanolab, generating new discoveries and innovative products that improve the lives of all living things.
So that gives a glimpse about the type of work we do. We'll now focus only on the poultry health alone. So the university has got about $77 million approximately for the Food for Thought program. We all understand and appreciate the need to enhance the productivity of food. By 2050, the population is going to be about 9 billion, which is just 30 years down the road. 2 billion people need to be fed with the same amount of resources we have today. So, which means the meat production is projected to increase by 50%, making the agricultural food as well as the livestock industry is putting in a key challenge. When we look at the egg as well as the poultry sector alone, approximately about 181 million tons of meat has to be produced from poultry, while about 102 million tons of egg need to be produced by 2050. So, which is about 1/3 to half of what we are producing today has to be enhanced and increased in terms of the production scale. And the current challenges are World Health Organization has recognized that there are 3 new diseases emerge every year. Out of these 3 new diseases, 2 are in the zoonotic area or the livestock area. So it could be in the poultry health as well as dairy and pork and other related livestock. Of these, 75% are zoonotic, which means there is a very high probability of the diseases transmitting from the animals to the humans. So it is a very challenging sector, challenging area to meet the current as well as the emerging challenges in terms of enhancing the food productivity, but also to sustain and to provide biosecurity to the whole agricultural sector at the global level. There is a paradigm shift is required. Right now, if a particular disease occurs in a farm or in a field, most probably samples are being collected. For example, if it's a poultry related avian influenza, oral swab or the fecal content or the litter or the blood droplet from the birds are being collected, shipped from the farm to the lab, such as the animal health laboratory here, to be able to assess what is the actual cause of the pandemic outbreak and what are the other additional parameters, such as what type of strain that is causing the disease. So the shift has to happen from moving away from shipping the samples to the lab and enabling the farmers to test, to enhance, to allow the farmers to make decisions in terms of health and also effective management strategies. We could also look at the agriculture, precision livestock farming from a holistic approach. When we look at the past 100 years, we have gone through 3 different revolutions. First revolution is mechanization, when James Watt introduced the steam engine just before the First World War, that created a huge productivity in terms of industrial products and manufacturing novel technologies, and also bringing down the amount of labor, physical labor that's being put in the farm as well as the other related sector. The second revolution is towards green revolution, enhancing the productivity of the amount of grain as well as milk and other related products from the agricultural farm. So scientific advances in livestock breeding can also be considered as one of the green revolution. The third revolution happened in the mid-1970s, which is about genetic engineering. It still happens. We all know the GMO foods and also the ability to resist a variety of diseases, both insect as well as pathogenic fungal diseases, and also overcoming climatic and as well as environmental related challenges in terms of enhancing the productivity. And then for the past decade or so, we are going through a 4th revolution in agriculture. We could call this as precision agriculture or precision livestock farming, PLF. So in this particular area, everything is related to time. Time accounts for money. Bringing down the time to results is one of the key factors in enabling the farmers towards making decisions. When we look at the poultry sector in Ontario alone, 41% of meat consumption accounts from poultry in the whole North America. Ontario's chicken industry contributes about $2.72 billion a year to the provincial economy, and also indirectly as well as directly leading to approximately 20,000 full-time equivalent jobs in the province. So it's a very significant effect in terms of managing effectively the poultry health and the other related livestock health. So the trends for future is shifting from reactive to proactive and also making a predictive approach. Even before the disease occurs, can we be able to understand subclinical signs just by tiny little biomarker at the very nanomolar, picomolar concentration in a much more non-invasive manner? Can we be able to predict the disease? So the whole game is enabling and moving from predictive to proactive towards predictive and preventive approaches. So there is a need for combination of in vitro as well as in vivo diagnostics to enable minimally invasive approaches towards diagnostic tool development. We all understand just a couple of months ago we had a influenza outbreak in the duck farm near St. Catharines. Last year during spring we had about 48,000 birds were euthanized all over the province of Ontario, which includes Woodstock here in Oxford County as well as in the other regions within the Southwest Ontario. So when we look at the economic aspects, billions of dollars have been lost globally, which is accounted towards avian influenza. But the key thing here is avian influenza does not occur frequently. They may occur probably once a decade or once in 8 years or so. Also, the possibility of loss of human is almost minuscule or negligible in North America, but it has happened in developing nations such as Africa as well as Asia. So these three different parameters and aspects has to be considered towards deployment of instrumentation as well as development of novel solutions for poultry health managing systems. So again, prevention starts with early diagnosis to move the farmers from predictive to proactive approach. So again, when we look at the avian influenza alone from a broader perspective, there are two types of strains, high pathogenic as well as low pathogenic. Low pathogenic does occur quite frequently while high pathogenic occur rarely. But farmers need to manage both the high pathogenic as well as low pathogenic to be able to make efficient management strategies in terms of enhancing the hygiene, in terms of providing sufficient feed or supplement to enhance the health of these birds. Currently, there are absolutely no pen site testing available. So farmers take a droplet of blood and ship it to lab. The lab typically RT-PCR is being used, which takes about 6 to 8 hours. So when the influenza outbreak that happened just a few months ago in St. Catharines in the duck farms, it took approximately 3 days for CFIA to come up with the type of strain to be able to determine what caused that particular strain. So the overall goal of the enabling and deploying instrumentation is to bring down the number of days, the time to results from 3 days to few minutes and also almost immediately. So that is the goal towards developing a handheld portable diagnostic tool to be able to rapidly detect the culturing as well as the identification of the subtype of the strains. We also need to look at the vaccine related problems. Vaccines sometimes does not work. There are some reasons behind it. So there are also several obstacles in developing successful vaccines. When we look at the hemagglutinin and neuraminidase glycoproteins, these are large targets of protective immune response. So vaccines are designed to induce hemagglutinin specific antibody response to protect the host from the infection. So there are 16 types of HA and 9 subtypes of neuraminidase. So if you create a permutation chart 16 by 9, you get a whole array of different combinations. So it's impossible to predict which particular HA or NA is going to cause that particular infection. So to be able to prevent vaccines, developing vaccine, a whole array of vaccine and also injecting a whole array of vaccine practically may not be feasible to prevent the particular disease. So inactivated avian influenza are also poorly immunogenic and also need very high concentration of hemagglutinin glycoprotein. So which means it's impossible almost or very difficult to predict the avian influenza specific virus which may possibly cross the species barrier and cause a pandemic outbreak. So although ideal vaccine would prevent infection, a realistic goal is to basically prevention through early diagnosis. So there is a bigger push in terms of the watchdog agencies, food inspection agencies, and also OIE, World Organization for Animal Health, in enhancing the real-time assessment of the health and also promoting towards biosecurity protocol. So by combining technologies such as geospatial technologies along with the handheld diagnostic technologies, it is possible to not only quickly diagnose even before the disease occurs, but also report the data in a real-time transmission manner to the end users, veterinarians, as well as the whole value chain to be able to make appropriate decisions. So again, the goal is to develop and enable rapid and on-time monitoring of diseases as well as the record keeping to meet that particular expectation. So from our lab, we have gone ahead and developed two technologies. One is a optical sensing technique. It is based on quenching. For example, if you run and then you feel thirsty, to be able to quench your thirst, you take a sip of water. So the quenching is directly proportional to the amount of the water that the body is being observed at a particular moment. So the same concept we use in nanotechnology using hybrid nanomaterials. Once we shine a light, the ability of the nanomaterials to bind to the proteins on the surface of the virus is being calculated in a real-time fashion. So because of the specificity and the affinity of a specific material to attach and bind to a specific protein, we would be able to distinguish and differentiate between two strains. For example, if the owner, the poultry farm owner, he or she has a specific flu and the birds are also going through specific flu, and if we need to detect and distinguish the actual strain in terms of identifying whether this flu for the particular farm owner is caused either by the human-based viral strain or the viral strain from the bird, it would be easy to distinguish right away by using this optical sensing technique. The second technique is an electrochemical technique. Electrochemical is typical to a handheld glucometer. So both the technique can be miniaturized. We have indeed using instrumentation approaches combined and integrated and interfaced with smartphone and demonstrated that it is possible to detect the presence or absence of pathogen and also distinguish between virus subtypes from just a droplet of blood sample that we collected from the pre-mixed sample in the lab. In terms of electrochemical approaches, maybe you might have used, or one of your household member might have used, diabetes testing. Just to prick a little finger and take a droplet of blood, put it in a little cartridge and stick the cartridge inside a handheld reader. The handheld reader would be able to immediately give the amount of glucose in that particular blood. So you would be able to make judgment in terms of changing the food habit or doing exercise or other related management decisions. Same concept, we have used an electrochemical approach along with nanomaterials, but focusing on the virus pathogen rather than the chemical biomarker to be able to identify, distinguish. We are also currently working on multiplexing. So if there are more than 6 to 7 types of strains are present, how do we go about screen all these viral pathogens? So we have the option to create and use an interdigitated technology, basically multiple fingers at a very microscopic level can be embedded and printed on a printed circuit board. The gap between the fingers, the height of the fingers provides a specific area. So the type of nanomaterial that is being coated on the interdigitated electrode has the ability to distinguish the specific viral strain. So because of the multiplexing and parallel processing approach, just a droplet of blood drawn from the inlet divides into 7 different streams, and each stream has different parallel arrays, and you can simultaneously detect what is the type of pathogens, subtypes of viral strains are present in that particular prototype. So this is the end vision of our technology. We have some proof of concept that we have developed in the lab. Still more work need to be done to be able to validate and also to test the point of care field usage related applications. So biosensors can also have a variety of potential. It can solve n number of problems. You name the problem, we would be able to solve the problem in one way or another using biosensors. This is an example in which a combination of, it's a wearable technology. You can actually put the tiny little loony sized sensor on the surface of the bird, either under the chicken wing, and you would be able to detect the movement, how fast or slow the particular bird moves. The same tiny little coin is also embedded with temperature sensor, so you could also detect the skin temperature of the bird and also specific biomarkers. So if there is a presence or increase or decrease in the amount of methane or other related biomarker in a real-time fashion. Along with that, it also has a tiny little Bluetooth module which emits and transmits the signal in a real-time fashion. So by combining this and putting together on the surface of the chicken, we would be able to monitor in a real-time fashion quickly or predict diseases. In this case, the experiments were done using a low pathogenic strain, CMKZ11 strain. So the chicken birds were placed in tiny little cages, and real-time monitoring of the temperature, the movement, acceleration due to gravity, as well as the particular odor were taken, and then in a real-time fashion the data was collected and analyzed. So examples of activities and body temperature along with the gas composition of the chicken infected with the low pathogenic strain. So we can, in a semi-quantitative manner, we can actually predict the actual movement of these particular bugs. So the question here is it possible, is it practically feasible to, if there are 10,000 birds in a little barn, can we be able to put a wearable technology on individual chicken and monitor all 10,000 chicken? Absolutely not. So there is a need for developing mathematical model and also a simulation to understand what is the possibility of efficiently deploying in terms of n number of particular sensors over n number of cages to be able to make the decision. So again, there are also companies such as Inomics, which is currently available, which allows real-time transmission of data such as carbon dioxide, methane, the temperature, as well as the sound, and the other related parameters to be able to assess the actual holistic conditions in which the particular bird is being grown over a period of time. So by combining the telemetry approach along with the individual bird related health factor, we would be able to make much more intelligent decision. So the whole concept of PLF, precision livestock farming or precision agriculture, is towards personalized individual animal health monitoring. So by understanding the actual differences and the heterogeneity associated between the birds, so specific management decisions can be made to enhance the welfare as well as the productivity of the birds. So this is an example of the topology in terms of deployment of the sensors. How many sensors one would need based on the area as well as based on the amount and the number of birds available in that particular zone. So Internet of Technology or Internet of Things is also another concept which comes under the umbrella of precision livestock farming. So to put it simply, transmitting real-time data related to health parameters using a combination of mobile phone and internet to enable the end users and also to monitor and track the different health-related parameters is the ultimate goal for precision livestock farming. So, in addition to the movement or the activity, there are other parameters can also be measured. One parameter is sound. It has been demonstrated that by measuring the cough of pigs, we could assess the health of the pigs, whether the pig is going through specific particular disease parameter. So, by taking into account the movement, the skin temperature, the gas composition, and also the vocalization of the birds, we could make a holistic as well as much more efficient decision on predicting the disease related parameters. So, here it is a population density management system. I am not sure whether you can see the picture here. There are tiny little bugs moving around. This concept was derived from chemotaxis. Chemo means chemical. So, taxi means going from point A to point B. There are quite a few taxis for bacteria, haptotaxis. They sense pH or gravity. Thermotaxis, they sense temperature. Similarly, at the macroscopic level for the birds do sense temperature as well as the, for example, if a particular amount of source of feed or the water is lacking, it has been demonstrated that the birds sense based on the movement of the other birds, and then they quickly move from that particular zone and try to go to where there is abundant source of food and feed and the water supply. So, the same concept can be applied by incorporating cameras at specific locations at the top level to be able to capture real-time transmission of the movement of the birds, to be able to assess the population density. So, population density can also be used as a factor in management of streamlining how much water has to be provided at specific zone within the poultry barn, as well as incorporating that with the health of the birds. Here is an example of the hybrid mobile environmental and the population density management system. And all these can not only be taken in real-time fashion, but also be transmitted using the IoT, and you can actually have visualization of how well the birds are doing in your smartphone at your in the palm of the owner of the farm itself. So, here is an example of an app that was developed by a group here. We can see here, these are very, very low cost approaches, primarily based on microelectronics and Texas Instrument based boards. These are about $80 to $90 each microcontroller board, along with few sensors. Overall cost is about $200. So, the screenshot of the app that was developed that provides information such as temperature, humidity, methane, the density value, and also the actual movement of the birds in terms of the grayscale picture. So, by using thresholding and image processing, we would be able to predict and make a very efficient decision right away. This is a framework that shows the possibility of deployment of wireless sensor nodes. Again, the question is, for practical application, wireless may not be easily available in a barn if the barn is away from a city or in remote places. So, that factor has to be considered in designing as well as the deployment. So, there are other Bluetooth related modules available to serve as a master as well as a slave module to be able to transmit as well as receive data at different locations. There are also recent challenges in terms of moving away from caged system to free range of the birds. So, in that case, then the whole design factor has to be considered in the deployment as well as assessing the automatic warning report towards the deployment of the monitoring system. So, event driven system for chicken health monitoring. So, by a collective assessment of all these parameters, then the trigger has to be sent through the mobile phone to the farmer to be able to make a judgment. So what are the factors one need to consider? Are these factors scientifically valid to be able to trigger that particular event to make a specific judgment? For example, if you have extremely high temperature of the bird as observed, and then your poultry litter composition in terms of methane is extremely high, do we need to call the vet or the Canadian Food Inspection Agency, because there is a possibility in terms of suspicion of the outbreak, because the factors correlate and fall within that particular threshold. So that factor has to have much more information, as well as assessment and mathematical modeling and stimulation has to be considered before scientifically validated to be able to deploy in real-time fashion. So most of these examples were done in a lab in a controlled environment. For practical usage, more experiments and more approaches are needed before realizing the technology in the farm. So this is another technology. It's basically a belt, a tiny little belt. So this is a blood flow monitoring technology, minimally invasive. So gentle poke on the surface of the skin on the chicken would allow the actual measurement of the blood flow using Doppler. So tiny little sound waves are continuously being passed, and then the actual reflection from the particular blood flow is being characterized, and we would be able to judge what is the actual flow of blood in that particular zone. In addition, we would be able to judge the composition of the blood, the biomarker, the protein, and the presence or absence of pathogens. So it's a little more intelligent in terms of providing the actual composition of the blood, but it's slightly invasive, which can also be practically feasible to be able to understand the health of the bird. So another example is chicken blood flow using Doppler flow meter. So the measurement principle and structure of the sensor probe has to be much smaller, so that the bird's welfare is not being sacrificed or affected in a negative manner. So this is an example that clearly shows the MEMS based technology that has been developed to be able to in a non-invasive manner. It's more of a wearable. For example, as I sweat here, as it gets warmer, I secrete specific salt composition that comes outside the epidermal layer of my skin. So similarly, the secretion of biomarkers on the animals can be measured in a non-invasive manner. We call this as tattoo technology. Basically put a sticker on the surface, and then the sticker would basically change colors. You shine a light on the sticker, and then based on the actual spectral information, spectroscopic change, you would be able to judge what am I secreting. Am I under stress? Am I secreting cortisol? Is my blood glucose level is changing? All this information can be easily assessed, measured in real-time manner, all in a much more non-invasive manner. Same concept for humans can be translated, either adapted or adopted for poultry health as well as other livestock, to be able to measure parameters in a much more intelligent fashion. This is a flock talk. So as pigs coughs, based on the cough, we could assess whether the pig is basically going through stress or hungry or specific physiological differences. Same concept can be adopted for poultry health as well. So there are a number of parameters we could assess just by the sound itself. Prediction of disease based on the tiny little differences. Identification of sex and genetic strain can also be measured using just by the vocalization of the chicken birds. Prediction of growth. So for example, the little chick as well as the mother bigger chicken, what is the difference in the sound? Some work has already been done in other universities such as Georgia as well as in Taiwan. And also we would be able to recognize the stress which would lead to enhancement of the welfare of the chicken bird just by measuring the sound. Again, when we incorporate and integrate this vocalization along with the activity in terms of the acceleration of movement, both gives a much more holistic assessment of the actual physiology and the welfare and the actual happening in terms of the feed intake of that particular bird. Here, an example was conducted towards a broiler vocalization for predicting disease. So, one of the major challenge in deployment and realization of this technology is noise. So, when we deploy this, fans are running in the poultry barn and people may use vacuum or something to clean. So, when we acquire all these signals, how do we go about subtracting the noise which are unwanted, but only look at the actual sound that's being generated by the birds? And what are the fine differences in terms of assessing the actual biological variables behind that particular sound? So, a lot more work need to be done to be able to realize and also deploy this technology towards having a vocalization technology for predicting disease. So, I would like to conclude by a video.
I am shopping with Mommy and Daddy today. I like shopping. Mommy normally lets me have sweets. I don't think Daddy likes shopping as much as I do. I think his job is to carry the shopping bags. Mommy and Daddy says that you have to be careful buying meat. That flag means it is safe. Farmers make sure their animals are safe and healthy. Birds can get sick as well. Some things that taste good can be bad for you. They can make some people get poor tummies. My mommy makes sure she gets food that won't make us sick, but keeps us healthy as well. Mommy has to be careful when she gives me treats. I can't eat peanuts. They make me go all red, and I can't breathe. But Mommy knows what sweets are okay for me. The best bit about shopping is eating all the food after. But Daddy says the best thing about shopping is getting back home.
Well, thank you so much for this opportunity. I would take questions or comments or suggestions at this time.
Thanks, Suresh. I thought it's a very fascinating technology. I wish I were a chicken and being experimented without being killed. So, Suresh can take questions if there are any. And may I ask you to repeat the question once asked, because we are kind of...
Sure, definitely, yeah.
So, yeah.
How close are you to commercialization of any of these biosensors for poultry?
So, how close are we towards commercialization of these biosensors that were developed in the lab? So, we have got some funding from the Griffin Layer program towards further validation and optimization of the biosensors. So, our current focus is primarily towards developing a portable handheld tool for rapid diagnosis of the and also distinguish the viral strain. So, we have made some advancements. Currently, we are working on the multiplexing as well as parallel processing towards identifying multiple strains from the same droplet of blood. So, there are some technological challenges associated to it. So, that's the stage we are in right now towards optimizing and enhancing towards detection of multiple strains. Yes.
The wearable devices that you can give to the chickens, do they take them off? Because I would think that would be something that they would like to do.
So, the mode of deployment is the question here. So, the question here is if we use a wearable technology, a sensor on the surface of the chicken birds, do they peck or do they tend to remove by itself? So, that still need to be addressed, but the sensor has the ability to provide assessment There are also non-touchable, non-invasive way of measuring. For example, when we incorporate video along with specific motion sensors. So if we incorporate 2 or 3 different video camera at different angles without actually putting any sensor on the chicken, we would be able to actually assess the gait movement, what is the weight and the impact of the bird, whether the bird likes the particular substrate in which it's walking around. All these can also be assessed, but that's a little more expensive because of the degrees of freedom involved and also other microelectronic components associated to it. In terms of wearable technology, yes, it has its own challenges. So the biocompatibility has to be considered when we design the actual wearability of that particular sensor to the skin of the bird's surface, how compatible it is. So the lighter the better. The material could possibly be made of much more biological in nature, so that they don't feel that there is a foreign object that's sticking to that particular surface. So these are other design considerations that need to be evaluated.
Suresh, I have a question. So in your antibody-based detection system that you showed, what's the source of your antibodies? I mean, are they monoclonal and?
Yes, some of them. We also currently been working on aptamer-based detection. So antibodies has their own advantages and disadvantages. So aptamers seems to be a little more efficient in terms of specificity. So we are also using aptamers, DNA-based aptamers, to be able to detect the pathogenic strains.
Okay. And that aptamers binding to the virus in the blood?
The surface proteins of the virus.
Well, if there are no more questions, please join me to thank Dr. Suresh for his time and excellent talk.
Thank you.